final exam. Whether it is internal assessment or finals, generally paper 1 and paper 2 will be there. So all those topics what you have in physiology will be split into 2. Okay. So now this is nervous system. This is like very big chapter what we have in physiology, from where you get lot of questions. Even essays also come from this chapter. Okay, from nervous system. So now let us talk about first 5 chapters maybe they have done. So I'll try to take these first 5 chapters now. So first thing is introduction to nervous system. So in this introduction to nervous system, basically nervous system is made up of what? Central nervous system and peripheral nervous system, isn't it? So central
Yes,
nervous
ma'am.
system is made up of brain and spinal cord. Okay. So in the brain also we have certain subdivisions, like we have forebrain, which is chiefly formed by cerebrum. Okay, then we have midbrain, and then we have hindbrain. 1 minute. Okay, but also the hindbrain, it has midbrain, pons, and medulla. Okay, and behind you are finding cerebellum. So you are see this whole like midbrain, pons, medulla. We generally call it as brain stem. The brain stem is now continuing as spinal cord. Okay, now. So whatever I have told you, the classification is given. Like brain, it has been divided into forebrain, midbrain, and hindbrain. The other terms you need not remember now. This is part of embryology. Then we can easily learn. Now not required also. Okay. So now just try to remember these names. Like whatever is given in the labelings, those are enough, Anjali. Okay. So this is only for your information. Central nervous system, and peripheral nervous system is made up of nerves coming from the brain and spinal cord. So how many pairs of nerves come from the brain?
Ma'am, 12.
Very good, 12 pairs. 12 pairs of cranial nerves we have. How many pairs of spinal nerves do we have?
Ma'am,
31 pairs.
31.
Yeah. Okay, 31 pairs. So that means the cranial nerves and spinal nerves will form peripheral nervous system, because though they start from brain and spinal cord, they go and distribute to various parts of our body. Okay. So like nervous system, see here, we have divided into CNS, that is central nervous system, which has brain, spinal cord. Peripheral nervous system, functionally, that is physiology wise, we divide it into 2, like autonomous nervous system and somatic nervous system. Okay. Under autonomous we have sympathetic and parasympathetic. This is the last chapter of this uh, uh, particular nervous system topic. But under nervous system chapter we have so many sub chapters, around 15 to 20 chapters are there. So one among them is autonomic nervous system. This itself is a separate topic we have. This also may come as an essay question, which I will be discussing later. Okay. Now, coming to the next chapter, or before I move to this, let me check if I have covered all important things or not. Persons listening, peripheral nervous system. Anyhow, no question will come from there. This is for your basic information, without which you will not understand following chapters. Okay. Now, coming to the next chapter, neuron. You should have studied in your schooling and all, like the typical neuron. How does it look? See, this is multipolar neuron, which is called as. So it has like cell body is there, and you have these projections which are called as dendrites. One big projection is there, which is axon. Okay, and in the cytoplasm you are finding the nucleus. We also find Nissel's granules, axon hillock. Usually on the axon you will find a sheath called as myelin sheath. Between the myelin sheath there will be node, there will be like nodes, no, which are called as nodes of Ranvier,
Ranvier.
which help in quick conduction of impulse. So there will be like millions of neurons are there in the brain, right? So all these neurons are actually connected from one neuron to another neuron. There will be a connection which is called as synapse. So functionally, the information will be moving from one neuron to another neuron via synapse. Okay, beta? So now, like that, that is the basic information of nerve cell or neuron, which is the structural and functional unit of nervous system. Okay. So neurons, now like when you just look at this, these are the processes which are called as dendrites. Okay, and a long process is axon. Now coming to classification, based on number of poles, we have classified them as unipolar neuron. That means it has only one pole, as you can see here. See, this is cell body, and from one pole you have dendrite coming and also axon coming. So if you have only one pole, we call them as unipolar neurons. Generally, we find these neurons during embryonic stages. Okay, then we have bipolar neurons. That means it has two poles. From one pole you have dendrites coming, and from other pole we have axons. So such bipolar neurons also we find in our body, especially in the retina of our eye. Rods and cones, they come under the category of bipolar neurons. And of course multipolar neurons, the most advanced and well developed neurons what we have exclusively in our nervous system. Full nervous system is made up of multipolar neurons. Okay. Now, depending upon the function also, we have classified neurons like motor neuron and sensory neuron. Motor is also called as efferent and sensory is called as afferent. Okay. Now when you talk about motor neuron, what do you mean by motor neuron? That means it is carrying the information from the brain,
Yeah.
like we have seen this picture previously. See,
From the
the
brain to
nerves
the
have started from the brain and it is going, like for example, you have like various nerves, like which nerve do you know? You tell me, based on that I'll tell.
vagus. Vagus.
Vagus, yeah, vagus. Vagus is one of the nerves. Which cranial nerve is it? 10th cranial.
10.
Yeah, very good. So that starts from the brainstem and it has extensive distribution. Like it is supplying the like most of the areas in the neck region and also thorax and abdomen. Okay. So this is parasympathetic nerve basically. So that means information is basically coming from the brain and the vagus nerve is carrying. Suppose if it is carrying information to the stomach, because that means nerve fibers are reaching stomach. So if the information comes to stomach, what happens? The stomach starts secreting gastric juice. Like that, those nerves which carry the information to from the brain to the peripheral organs, you call such nerves as motor nerves. Okay, also called as afferent nerves. Motor are efferent, sorry. Then we have sensory. What is the meaning of sensory? That means
From the, I mean, the path to the brain. They
from,
convey the
from where? You're right.
to the to the brain.
To the brain is correct, but from where it starts?
Ma'am, from the sensory organs or skeletal
From the peripheral part. Okay. It can be
muscle.
skin, it can be an organ. That means it is carrying information. Suppose if you are touching a hot object, what you will do? Immediately you will withdraw your hand. That means
Withdraw.
sensory nerves are carrying the information. Okay, from the peripheral areas, the information is moving to brain. So such neurons are called as afferent neurons, also called as sensory neurons. Okay, just remember these words very carefully. Now, here you are finding the beautiful picture of neuron, where you are finding, see, this is the cell body, cytoplasm is seen, so many granules are seen which are called as Nissel's granules. These projections are called as dendrites. Okay, and one big projection is axon. The place where axon starts, you call it as axon hillock. Okay, and see in the axon cylinder we are finding these are all myelin sheaths. In between, you are finding the nodes which are nodes of Ranvier. Okay. Then coming to depending upon the length of axon. So that means this length of axon can be as big as starting from the brain till moving to stomach, as what you said. Like for example, you said vagus. So the length is more, you call them as Golgi type 1, and if the length is less, you call it as Golgi type 2. Just read this once. So Golgi type 1 neurons have long axons, isn't it? Cell
Yes.
bodies are present in the brain and their axons reach to remote peripheral organs like stomach. Cell body, suppose if it is present in brain stem, it is moving. So such long neurons are Golgi type 1. Type 2, short axons. Okay, that means from the brain they start, or from the spinal cord they may start and they are ending there itself. Short neurons. They will have connections through synapse. Multisynaptic, so many connections. Okay, then structure of neuron we have just discussed, like it has nerve cell body, dendrites. Okay, then so coverings of neurons. We have seen myelin sheath, right? So like when you see the bundle of neuron also, like you have, like you were saying, like we have epimysium, perimysium, endomysium. These were the terms they were used to describe the bundle of muscle fibers. Similarly, we have certain coverings. Suppose there is a cover which is immediately covering one particular neuron, you call it as endoneurium. Bundles of nerve fibers, they are all covered by perineurium. And whole of the nerve, big fiber, like whole of the nerve is covered by outermost covering which is called as epineurium. So this is the cross section of nerve. Okay, individual nerve fiber covering is endoneurium. One bundle of nerve fiber is covered by what? Perineurium. And
Thorium.
all of the nerve is covered by a covering which is called as epineurium. Okay, so these are the coverings. Internal structure is like this. So this is the axon cylinder over which we have myelin sheath, and the myelin sheath is secreted by a special cell called as Schwann cell. Between the myelin sheath you have nodes of Ranvier, okay, over which the impulses will be jumping. So functions, as you said, like myelin sheath helps in faster conduction of impulse. Insulation, in the sense, it is something like electric current which is moving from one neuron to another neuron. So if sheath is not there, the electricity will spread like anything. There will be so many shocks like, okay. So like in a crude language, I'm telling you. So that means it has insulating capacity which prevents the unnecessary stimulation of impulses all around, so that the impulse is going in a particular direction. Okay, so the Neuralema is the thin membrane like this, as you are finding. This is the axon cylinder, and over which you have a covering which is actually called as Neuralema. Okay, a sheath of Schwann. So the difference between myelinated and both type of nerves we find in our CNS, but myelinated ones are only more, and that is the reason our impulses are moving very fast. Okay, that means it helps in quick conduction of impulses. Then neurotrophins. These are protein substances which are playing an important role in growth and functioning of nervous tissue. So that means if there is any degeneration in the nerve fiber, you will see they are again regenerated because of the release of these like chemical substances which are called as neurotrophins. Okay. Source, like generally many tissues of our body will release like in muscles. Next chapter will be neuroglia. You will be coming across these terms, astrocytes and neurons. Okay. So neurotrophins, they facilitate the growth and development of nerve cells. They also help in repair mechanism. They help in the maintenance of nervous tissue and neural transmission. So suppose if these neurotrophins are not released, then usually you will see at certain age, old people, they may have like long, like old memories still they remember, but new memories they usually forget. So that is basically called as like Alzheimer's disease, forgetfulness. Okay, because you will see as the age advances, the neurotrophin release will be reduced. Okay, and the muscle tone also will be reduced, and it will lead to a disease called as Parkinson's disease. This details of this disease we will learn when we do basal ganglia chapter in this nervous system.
Yeah.
Okay,
My Alzheimer's is like they're almost the same.
sorry, both are different.
Parkinson.
Both are different. Parkinson's disease occurs due to, as you have studied few neurotransmitters in college, one of the neurotransmitters name was dopamine. So suppose if dopamine is not synthesized, then you will see the neuroinhibition doesn't occur and there is continuous stimulation only. So that you will learn as you proceed in a proper systematic way. Okay, I'll tell you. Parkinson's itself is a separate topic when we do basal ganglia. Okay, for now you try to remember due to the deficiency of neurotransmitter like dopamine, usually people suffer with Parkinson's disease. Whereas Alzheimer's is forgetfulness, loss of memory. Okay, generally is seen in old age. Okay, then so neurotropins, whenever they are synthesized, they will have certain receptors. Neurotropins will go and bind to those receptors, and that's how that particular area will show the effect. So that is mode of action. Anyhow, neurotropins and all are not required, but still, to complete the topic, I'm telling you, even nerve growth factor, all these are not required. And in case if you want to study, also just try to remember the names. Neurotropins, what are the different names you have, and few functions. Okay, but this is only for general information. Now classification of nerve fibers is very important. Okay, classification till now whatever we have read, classification of neurons is important. Okay, and what else did we study? And that types of nerve fibers like Golgi type 1, Golgi type 2, that is also very important. Okay, length of, based on the length of axon, this also usually will come for your final exam. Types of neurons and classification based on length of axon, rest are only for your general information. Okay, now coming to class, this itself can come as a 5 marks question, this chapter, classification of nerve fibers.
Okay.
This is quite simple also. Now
Ma'am, how do these exams work, ma'am? I mean, the first year exam. Uh, is it conducted by the college itself?
internal assessment exams will be conducted by college, and the university exam will be conducted by the university only. Question paper will be coming from the university. And nowadays, even internal assessment paper also is sent by university only, because all colleges are
Ma'am,
following
you
uniform.
mean which university?
KNR, Kaloji Rao.
Oh, right. Yes, ma'am.
Kaloji
Right.
Rao. We have this Kaloji Narayana KNR University. You are from Mamta Medical College, right? So
Yes,
our
ma'am.
Telangana state is full, it is affiliated to KNR only. Okay,
Yes, ma'am.
KNR UHS. So now coming to the classification of nerve fibers, like we have, depending upon the structure, like we have myelinated nerve fibers and non-myelin. This is very simple, we know. If myelin sheath is present, you call myelinated. If it is not present, you call it as non-myelinated. Based on the distribution, like somatic nerve fibers, if they go and supply the skeletal muscle, you call them as somatic. As you said, vagus. I said it goes and supplies the stomach. That means if it goes and supplies the internal organs, you call them as visceral or autonomic nerve fibers.
Nervous.
Now, based on the origin, if they start from brain, you call them as cranial nerve fibers. If they start from spinal cord, we call them as spinal nerve fibers. Now, depending upon function, like sensory nerve fibers will carry the information from the sensory receptors to the brain, isn't it? So those are afferent fibers. And if they start from the brain and then go to peripheral areas, we call them as motor nerve fibers. Okay. Now, based on what neurotransmitter is released at the nerve ending, again we have classified them as adrenergic and cholinergic. If the
Energy.
neurotransmitter is noradrenaline, we call it as adrenergic.
Adrenaline.
If the neurotransmitter is acetylcholine, we call them as cholinergic nerve fibers. Okay, and based on the diameter, this is very very important. Based on the diameter of nerve fibers, again we have classified them into alpha, beta, gamma, delta, B, and C. This is called as Erlanger-Gazer classification. This question comes pakka usually. Directly they won't ask you, like there will be a question like Erlanger-Gazer's classification. Then you have to just write this table, like based on the like diameter of the nerve fibers, thickness of the nerve fibers, you will see conduction of impulse. The thicker the nerve fiber, the faster the conduction. That you remember. Okay. Now see here, alpha, the diameter of this nerve fiber is how much? 12 to 24 microns. How fast is this? 70 to 120 meters per second. As you move on, alpha, beta, gamma, delta, B, and C, the diameter is reducing. The C1 is less than 1.5 microns, and you can see the speed, it's so less. The
Yes.
velocity is 0.5 to 2 meters per second. Okay, so give this table for this Erlanger gases classification. Okay, better. So that finishes this chapter. Then coming to properties. Properties is the one what you was dealt in your college also, isn't it?
Yes, ma'am.
Now, under properties of, see this, definitely this is also a very much expected question which comes for 5 marks. Okay.
Yes, ma'am.
Now, let us see what are the various properties we have. First thing is excitability. One thing I told you, when we touch any hot object, what do? We do just withdraw our hand. That means what? Our nerve fibers are highly excitable. So this is physiochemical change that occurs in tissue when stimulus is applied. Like hot object is a stimulus. Okay. So that is an external agent which produces excitability in tissues. Okay. So this stimulus and strength duration curve now we will see in now anyhow this is uh, 30th chapter in the sense that is muscle physiology. Okay, chronaxy and all that details we will learn when we do muscle physiology. Okay, but whatever are related points I will tell you, nothing to worry. Okay, so
Yes.
response due to that's the reason basically we have to start with muscle first, then come to nerve. But they have
Yes, ma'am.
started with nerve first, pakka.
Ma'am,
They have started with muscle chapter or nerve chapter in your college, nervous system.
ma'am, actually, what is happening is that things are happening simultaneously, ma'am. So we're not able to correlate it. We are having like 2, 3 tracks for physiology, 2, 3 tracks for anatomy.
Okay,
So
just
it's all.
write the topic. What is the topic which is the daily? Just give me the
Yeah,
name.
ma'am, muscle they started this. They were talking about what kind of muscles are there. Like one page I told you, no, ma'am, striated, non-striated. They told us that, and then they immediately went off to the next chapter.
Which chapter?
Fascia and
Ayo,
is it?
Anjali, you are
No, that is
mixing
anatomy, right?
physiology with anatomy. It's okay.
Ma'am, yeah,
It's
yeah.
okay. Okay. Today we are
But
doing physiology. Don't mix up with anatomy. Huh?
we're not able. find it out.
It's okay, it's okay. You are only in the beginning, no? No problem. And we are learning
But no,
more than.
muscle was started with anatomy. That is why I am telling you. They didn't start muscle in physiology. They started muscle in anatomy. That is when they went to skin and fascia.
That is all general. Okay?
Hmm.
Under general topic we have. Oh, just a minute, I remember something. We were doing excitability, right? So nerve fibers have low threshold for excitation than other cells. Means they immediately respond to some stimulus. That is the meaning. So response due to stimulation of nerve fiber. So based on the strength of stimulus, like if you just take a current shock, like low voltage, high voltage, something like that, it would be, isn't it? So like that. So action potential develops. But before that action potential curve, you need to understand better. Resting membrane potential action. This all is explained in muscle. So okay, I'll take you to muscle and then come back. Otherwise, you will not understand. Remember this page number. Make a note. 769.
Yes, ma'am.
This is the chapter which you should understand first in order to understand the nerve properties. So, what are the changes we see in muscle contraction? So, whatever related things I will tell you. So, first of all, we will go with the resting membrane potential. The resting membrane potential is the potential which is actually, we will see the definition first and then I will tell you the details. See, electrical potential difference across the cell membrane. It can be cell membrane of a neuron or it can be a cell membrane of special senses like rods and cones and also like hair cells of the ear also. This is applicable for many things. Cell membrane of a skeletal muscle. So that means a potential difference change across the cell membrane under resting condition is called as resting membrane potential. Okay. So there should be certain ionic changes for this muscle fiber or nerve fiber to get excited. So it's shown like in a very simple and easy way. So development of resting membrane potential by sodium potassium pump. See, this is suppose now because we are doing neurophysiology, you have to think it is a neuron only. So and this is the cell membrane of the neuron. So we are finding sodium, like suppose if the impulse comes, like there is some, like hot object is one impulse, like there is some impulse because of which you will see ionic changes will be taking place. So what will happen? Sodium potassium ATPase enzyme comes and it will cause ionic exchange. What is that? 3 sodium ions will go out. In exchange, you are finding 2 potassium ions are coming inside. That means positivity is more outside, isn't it? 3 plus ions are out and 2 plus ions are in. That means more positivity outside. Comparatively, you call it as negativity only inside. So because of which, what happens? Action potential develops. Got it? Read this. See, development of resting membrane potential by sodium potassium pump. You are finding diffusion of ions, that is, like you are finding sodium outside. Like how many sodium ions are going outside? 3. How many potassium?
Three,
Yeah, 2.
two,
Okay, so you are finding like potassium leak channels are opened and many of the sodium leak channels are closed. So this is resting membrane potential. So if such ionic changes are taking place, what happens is, suppose this is skeletal muscle. So you are finding initially this skeletal muscle was at rest. Okay, suppose the muscles of palm only, they were at rest. Okay, now because you have touched the hot object, there were ionic changes, that means changes in the resting membrane potential. So what happens, skeletal muscle will contract and withdraw your hand. What is happening, see here, before zero, whatever potential which is ionic changes which are taking place, we are calling it as resting membrane potential. So now once the ionic, see this area A, opening a few sodium channels. So what is happening, like once the stimulus is there, you will see the skeletal muscle starts contracting, and this is the
Yes.
peak of contraction. This is applicable for nerve fiber also. The nerve fiber also is excited and it goes to the peak area of its excitation. Now everything, whatever, see skeletal muscle, do you think it is always contracting or is it also relaxing? Contraction.
Ma'am, sometimes contracting, sometimes relax.
Yeah, once you contract, you also relax, isn't it? That
Yes.
means contraction followed by relaxation. So whichever you are finding the graph going up, you call it as depolarization. It's written here, you see, depolarization. Okay, so the time period is 0 to almost 1 millisecond. You will see the neuron or the skeletal muscle, it just shoots up. You call this graph where either the muscle or the nerve fiber is excited, you call it as depolarization. After some time, what happens again, sodium ions will come inside, potassium ions will go outside. So what happens, the nerve fiber will relax. It is, you can see the graph coming down and this whole blue area, what do we call it as, repolarization. Okay, so repolarization is further subdivided into after depolarization, after hyperpolarization and all. Okay, so that is the action potential curve. Now you have to correlate this information. Only thing is, whatever millivolts we have here, like depolarization action potential for, in case of skeletal muscle fiber, it is 7 to 10 millivolts. So in the form of table it is given there. I will take you to the table and now you will understand. What's the page number?
769.
7.
769, you said.
Okay, now see here, action potential or nerve impulse. It is similar to that of muscle, just now what I have shown you. Few minor differences, in the sense only the readings will differ, but the concept remains same. Okay, beta? Now see,
Yes.
resting membrane potential. In case of nerve fiber, it is minus 70 millivolts. If it is skeletal muscle, it is minus 90. Okay, firing level. See, same words are used. If you see, this is amplitude, that is on y-axis, and this is time period on x-axis. You can see time period is 0, 0.1, 0.1, 0.2, 0.3, and 0.4. Because nerve fibers are very fast when you compare with muscle. You have used the word 1, 2, 3 over there, but milliseconds. Here in 0.1, 0.2 you are using. See, this is stimulus artifact and this is depolarization, what I said. And this is repolarization or after polarization. Okay, so firing level, how much is the action potential, all these values you try to read and just read once, you will understand. I'll send the link of this book. Make sure you read all these 5 chapters what I am teaching you. And if you don't understand anything, tomorrow we will discuss. Okay,
Yes.
so that is action potential or nerve impulse. Okay, so then we have electronic potential or local potential. So non-propagated local response that develops in nerve fiber. That means it remains there only. It's not moving from one neuron to another neuron. Subliminal, that means minimal stimulus, that is. Okay? That does not
Yes.
produce any action potential. You call it as local potential. Okay? Or graded potential. Then properties, it is not propagated. Basically, if one nerve fiber is activated, all the neurons one by one,
All of
that's what is all in none, but this electronic potential is not following that. Okay. Then we have voltage clamping. That means we are making use of certain electrodes to prove like action potential is generated, and you will see nerve fiber is moving in this way and all. Okay, so voltage clamping refers to experimental method that uses electrodes to alter and control the action potential, membrane potential, resting membrane potential. Okay,
Yes.
so again the details will be discussed in another chapter when we will be doing muscle beta. Just few
Yes.
points try to read once. Okay. Mm. More than anything in this chapter, you should know this one, saltatory conduction. I will tell you before that. See, conductivity. What is the meaning? It's an ability of the nerve fiber to transmit impulses. I said
Yes, ma'am.
we have millions of neurons and long nerve fibers. It can be Golgi type 1 or Golgi type 2 neurons. If they are Golgi type 2, information from one neuron to another neuron should move continuously, isn't it? So that
Yes.
mechanism is basically through conduction. Okay, so mechanism of conduction of action potential. So same thing better like depolarization followed by repolarization. I hope these two terms are clear now. Okay,
Yes,
go to that
ma'am.
chapter also, then you will understand more clearly. Now, what is this saltatory conduction? Very simple. See here, you are finding this is non-myelinated nerve fiber, and this is myelinated nerve fiber you are finding. So, if an impulse has come to the non-myelinated nerve fiber, of course the information is moving from the axon hillock, it is trying to move to another neuron, but you can see the speed, how slow it is moving. Okay, so that is whereas in case of myelinated nerve fiber, where you have this myelin sheath, these are the nodes of Ranvier, you are finding the impulse is jumping from one node to next node, from this node to this node, and so on. That means faster conduction of impulse is called saltatory conduction, where you are finding the impulse is jumping from one node to another node. Okay, better? Then next property we have is refractory period. That means what? Resting period. The nerve fiber is nicely taking rest. It is not responding to any impulse. You call that period as refractory period. Absolute refractory. How much ever stimulus you are giving to the nerve fiber? No, I won't respond at all. The nerve fiber is telling like that. You call it as absolute refractory period. Relative refractory period in the sense, to some extent, if you give an impulse, more or less a small response is elicited. Then you call it as relative. See, period during which the nerve fiber shows response, the strength of stimulus is increased to maximum. You call it as like relative
Yes.
refractory period. Summation. See, when we do, like there is a chapter called as synapse, where nice pictures are given to understand what is summation. Okay, for now, just try to understand the meaning. That means once subliminal stimulus, I said it local response, no response is subliminal, isn't it? It doesn't produce any response. Okay. However, if two or more subliminal stimuli are given simultaneously, small response is elicited. That's what is called as summation. So suppose if the subliminal stimulus what you are giving is only one, so, but such subliminal stimuli you have given 5, 1 plus 1 plus 1 plus 1 plus 1, how much? 5, no? So that means that
Yes.
will be enough to elicit a small response. That's what is called as summation. Okay. Adaptation. Adaptation means what? Suppose now formalin initially you should have felt on day one, abba, it is so like powerful and it is causing giddiness, watering of eyes and something like it was looking like an irritant, and now you are more worried about like what is being dissected, are you able to see the nerve, are you able to see the muscle or not. That means what? You got used to that. That means our
Yes.
nerve fibers also have the property to get adjusted to certain things. That's what is called as
Yes.
accommodation. Okay, that means our nerve fibers are stimulated continuously. Depolarization is also occurring continuously. Okay, so that sodium pump is getting inactivated and our body is adjusting. Okay, now infatigability, what does it mean? Nerve fibers cannot be fatigued. That means they are not tired. Okay,
Yes,
it is stimulated continuously for a long time. Okay, so that means If they are all right, they will conduct impulse. Otherwise, they will just keep quiet. So that's the reason.
Yes.
Okay, that is. That's what I said. Absolute refractory period. How much ever powerful stimulus you give, it's not responding, isn't it? Once it has
Yes.
completely got like relaxed, then only it is ready for the next impulse. Okay. All or none now. When the nerve fiber is stimulated by a stimulus, it gives maximum response or doesn't give response at all. Okay, that is all or none now. So, is this also over? Degeneration, regeneration?
Yes, ma'am. They just started it today.
Okay. See, what do you mean by this degeneration and regeneration? So, suppose if a nerve fiber is cut, see here, Anjali, this is axon cylinder, and you are finding this nerve fiber is cut due to some injury, road accident, or something has happened. So, what happens is the dendrites, just compare the structure of the cell body of neuron. The dendrites are disappearing, Nissen's granules are disappearing. You are finding nucleus is pushed to one corner, and you are finding myelin sheath is disappearing, isn't it? Distal to the injury, you are finding these changes, and you are finding breakdown of this axocylinder. But after few months, like 6 months, 8 months, because nerve fibers do not heal fast, regeneration is very poor in neurons. Okay, that's the reason in paralysis cases people do not recover fast. Okay, so injured nerve fibers you are finding they are gradually regenerating. As if there is any cut in your body, after few days you will see healing will occur. So joining of both cut ends, okay, beta, then regeneration is completed. As the first neuron only it looks, the last one also. Okay, so myelin sheath and Nissen's granules have come back. So this type of degeneration is called as Valerian degeneration, also called as arthrograde degeneration. This is the only very important topic what we have in this chapter. Okay. And remaining, just try to give one reading, and this textbook is very good. In fact, you will easily
Yes.
understand. Okay. So I'm stopping here. The remaining topics we will discuss in next class.